All right, Presley. So let's talk about 6-2. This is the structural drawing, and let's talk about what a glucose structure is. The glucose, that's blood sugar, and so here's how you draw a glucose. Glucose is C6H12O6. That's what you would have learned it in biology class. And remember, that's its empirical chemical formula. Its structure will be a ring. And since it's a hexagon because it's a 6-part ring. So you draw yourself a hexagon. This corner here is where the oxygen is. If you remember from the ribose from the past, there's always an oxygen in this ring, and by convention we put that oxygen up and to the right. So then this is first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sugars will have that hand sticking up for their last carbon. So hopefully you can kind of remember that from the ribose. All right, then we've got OHs and H's that go around. So with the glucose molecule coming off the first carbon, we have an OH, and then down here is an H. And then these alternate. If OH is on top, we got an OH down here and an H, and then we got an OH up here and an H, and then an OH down here and an H, and then we alternate to an H here. So that's, if you can remember to put that OH at the top there, then the OHs just alternate with the hydrogens. Last thing, we've got an H here and an H here and an OH on top. So that's going to be the same as the ribose structure. And therefore, if we add all of these up, we should have 6 carbons according to the empirical formula. 1, 2, 3, 4, 5, 6. Check. We should have 12 hydrogens. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And then we should have 6 oxygens. 1, 2, 3, 4, 5, and then don't forget this one, and 6. So yeah, so all of these atoms are there, and this would be their actual structure. Okay, so that's a glucose. Pretty much just like the ribose, just with one more carbon. And since the ribose was a pentagon, one more carbon would mean now you'd be making a hexagon. Okay, so glucoses, when we eat them, we are rarely just eating a glucose molecule. We're eating a polymer of glucose, a carbohydrate. And a carbohydrate is going to be a long chain of of glucose, such as starch. And then during digestion, the chemicals in our stomach break those polymers down into monomers, and those monomers are absorbed into our bloodstream. So let me show you how these are hooked up. How does a polymer work? All right, so let's just put 2 glucoses up here, and then I'll show you how they're bonded together. All right, so I'm just going to draw another glucose. In the previous unit, when we did riboses, we put them vertically because that's how DNA is built, and that's by convention that just works well when you're talking about DNA. Well, with glucose and starch, it's horizontal, not vertical. So I'm gonna draw another glucose here. alternating okay, so there, 2 glucoses. Here's how they would be hooked up. And so plants would do this. Plants make the glucose from photosynthesis, and then they make the starch. Here, I'm going to shut the door. Mr. Richardson's class is dissecting turtles. All right, so here's how a plant would initially build the starch. It would make the glucose from photosynthesis, and then what it's going to do is it is going to come here. We're going to go to this carbon, and we're going to clip off just the H. So all you have is an O there with a dangling bond, and then the machinery of the cell clips off that entire OH group. So what you end up with is an H clipped off there and an OH clipped off there. Those react to form water. And so you have a dangling bond down here, a dangling bond there, and so these bonds can be hooked up. I just draw it like this. It's a diagonal. Okay, so that's how 2 glucoses are hooked up to form a very small starch, and then this would just go on and on and on and on, and the starch molecules get huge. All right, the name of this bond, this is called a 1,4 dash glycosidic linkage, where 1 and 4 refer to the carbons in the bond. This is the first carbon, 4th carbon, and glycosidic just means, I don't know, ancient language, glycosidic just meant sugar. So this is a linkage between 2 sugars connecting the first carbon to the 4th carbon. All right, so that is how glucoses are polymerized. Now, let's talk about amino acids. When it comes to an amino acid structure, a single amino acid, here's how you draw one. You start with a carbon and you put 4 bonds off the carbon. Off to the left is a nitrogen with 2 hydrogens off of it, and it's perfectly fine to draw those diagonal. On top here is a hydrogen. On the right is this group. It's a carbon double bonded to an oxygen, bonded to an OH. And then down here is a question mark. There are many different amino acids, and they all have this structure, but they vary at that point. And so I'm not talking about one specific amino acid here. I'm just giving you the general structure. So we're just going to put a question mark there. You would put a question mark there, and just leave it until we talk about specific amino acids. Then we'll fill that in. But we're not going to do that in this unit. All right, so that's an amino acid. The reason why it's called an amino acid is because in chemistry, we learned about a molecule called ammonia, which looks like this in 2D. And this right here is an ammonia. It's just that this H has been clipped off, and then this group has been hooked to the carbon. So when you do that, in the biochemistry world, this is now called an amino group to reflect its ammonia origins. This group over here is an acid group. That's because in chemistry you learned an acid is a proton donor. and this is a proton, and it gets donated. Therefore, this whole structure is known as an acid group. All right, so the reason why this molecule is called an amino acid is because it's literally from left to right, amino acid. Okay, so how are amino acids polymerized? All right, a polymer of amino acids is known as a protein. So let me show you how these are hooked up. So what I'm going to do, just like in the glucose molecule, we're going to go sideways, not vertical. I'm going to draw another amino acid here. Okay? And what the machinery of the cell does when it connects amino acids together is it clips off this OH group. This is a theme. Every time we've done this between the nucleotides, and then you just saw the glucose and now this, it's always clipping off an OH somewhere, and then clipping off an H somewhere, and then those go on to be water. So one of these hydrogens will go, it doesn't matter which one, I just pulled the bottom one off. And what we'll have is this dangling bond is connected to this dangling bond. And so now we have this connection. This is known as a peptide bond. Not sure why it's called peptide. All right, so then that would just be repeated over and over and over again for a cell that's building proteins. And that could be an animal cell, that could be any cell. Whereas with the glucose, plants do that, photosynthetic organisms do that. This could be done in any organism. All right, now how this fits with this unit is you receive polymers from food that you eat, and those have to be broken down into their monomers. So what digestion is doing is it's breaking these peptide bonds, so you end up with just the amino acids, or it's breaking the glycosidic linkage, so you end up with just glucose. It would be breaking the phosphodiester bonds, so you would end up with nucleotides if we brought that in from the previous unit. All right, now I'm not going to have you worry about the structure of fats. Don't worry about that. The important fat part will be in the next target 6-4. All right, so see you later.